Hello everyone, I am Pankaj Kumar and today we are going to learn about the DNA library. As you know, library means something which is consist of number of books in for the different disciplines, and for different discipline it is kept safely. Whenever you want to read recombinant DNA technology, you will go and get one genetic book. Whenever you want to read the structure of the glucose and the bonds in the glucose, you will go and bring the Leninger book of the biochemistry from your library. Like that, we have a DNA library. As you know, we have how much? Okay, we okay. Everything is fine. No, yes, fine. It is. So we have around 6.6 10 power 9. This much on nitrogenous basis. Okay. So we want this will be equal to 46 chromosome. Okay, so you have a different chromosome for the different disorders, and suppose you want to know the Down syndrome, then 21 chromosome number, you want to extract the 21 chromosome number from any organism. So where you can store this 21 chromosome number? You cannot cut and get the 21 chromosome number every time from a human cell. So what we will do, we will split the complete 46 chromosome in a different manner, and we will make a library like 21 chromosome is there in that X box, and then 23 is there in the Y box, and then 20 is in the Z box. Okay, like that you made one shelf and you put your DNA. Now, what is our shelf? Shelves will be our vector. Hopes you understand. That is a library of the DNA in which you can carry your important details or import the sequence of the nitrogenous bases. You can put it there. Understood? So we have two types of library. You can see genomic library and a cDNA library. Genomic the library encompassing of complete genome. Like if you have 46 chromosome, then all the 46 chromosome in this 46 chromosome you have a two important portions. What are the important portions? The heterochromatin, and euchromatin. What it is? Euchromatin. So heterochromatin which not express. Okay, not expressing, not expressing. And this euchromatin is what? Expressing. Okay. Euchromatin is expressing gene. So in the genomic library, you'll make the library of complete genome. You will not distinguish whether it is a heterochromatin, whether it is a euchromatin, whether it is a intron, whether it is an exon. You will not distinguish accordingly. But what if if I will ask you to bring the mRNA only, or to bring the expressing gene. If I will ask to go Mohan and bring this euchromatic library. So what you will do? I will explain this with the one example. Suppose you went in a library. So in library, 1,000 books are there. Okay, but if I will ask you go and bring the class 12th book of the library in the library, then you will ask, would you like to take NCRT book or GRB book or Arihant book or which book? Then I will ask you to bring the NCRT book, class 12th biology NCRT book. So for that we have a separate library. Okay, understood? But if I will ask go and bring the book biology, then there if I will ask you to go and bring the book from the library, it means it consists of complete books, physics, chemistry, math, biology, etc., etc., etc. That is our genomic library. Okay, in which every book is there. Okay, every genes, every sequence of the DNA is there. Whether it is required, whether it is not required, it is there. Okay, So the definition is that in it encompassing the whole genome, that is called as genomic library. How we can make a library of this DNA? Is it possible to make a library of a DNA? Yes, it is. How you will make? What you will do? You will take the DNA and then fragment with the restriction endonucleases. Once you will fragment it into the several fragmentation, then what you have to do? You have to keep took this A, B, C, D, X, Y, Z. So many fragments will be there, and then you will insert in your vector. After inserting in a vector, then you will clone it, and you will make a clone as a bacterial colony, and then you will grow for the several years, several months, several years, like that. And whenever you want bDNA, then you will take this bDNA from this specific bacteria, and then you will isolate this specific fragment, B fragment. Understood? So you can see that how we can make a DNA library. But if anybody will ask you how many clones you have to make for the 46 chromosome, okay, or for the 46 billion nitrogenous base. Suppose somebody asks, there is a 4.6 billion nitrogenous base are there, okay, nitrogenous base are there. How many clone you can make? Okay, how many clone? Then you have to ask how many fragment will be there. Okay, then you will ask, sir, how many fragments are there? Then for that only you will accordingly you will make a clone only, na. If we have a four fragment A, B, C, D, then you will make four vectors or four clones. Got it? But you have a 4.6 nitrogenous bases. For that we cannot do the simple mathematics. For that there is a one formula. I would like to tell you that formula is n is equal to ln 1 minus, this is a probability of a this is a probability of the clones, and then log 1 minus a by b. This is your formula. Here n is a number of clone. Okay, what it is? Number of clone. And then p is a probability of a fragment in a DNA. Okay, probability of fragment in DNA. Okay, of fragment in DNA. Okay, then a is what? A is the average size of a DNA fragment inserted into the vector. Okay, average size, like A will be, suppose A will be how much? 6 base pair it can be, okay, and B will be four base pairs, like that. That is the average size we will take the fragment DNA. So A is a average size of DNA fragment, DNA, we can write inserting DNA fragment, okay, DNA fragment. And then one more is there, that is B. Then what it is B? You can write B is a total size of the genome, okay, total size of genome. So you can solve this question if 3.3 billions are there, average size of the fragment is around around three base pairs. Sorry, not three, three, 3 kilo base pairs. Suppose, then how many clones you can make? The probability of the fragment in a DNA will be around 3.3 means billions means you can make suppose 100 fragments. Randomly, I am giving. You can tell your answer in your comments. Okay, and I will tell you the correct answer there only. So thanks for giving your answer in the comment. Now we'll see the second library that is called as Cdna library. This C means what? Cat, not cat. Okay, it's not a cat. It's a complementary DNA. Got it? So complementary DNA means some. It is not exactly DNA, but it will complement the DNA. So how we can make a DNA without a DNA? That is called reverse transcriptase. Okay, you can see here there is a reverse transcriptase which will convert the mRNA into complementary cDNA. How it will convert, we don't know. This reverse transcriptase enzyme will help in the formation of a DNA strand. Instead of RNTP, it will add DNTP in the strand and then it will make a DNA. So mRNA is a tissue specific. Like the question is that how we make a library of cDNA and why we need a cDNA. Okay, we already have a genomic library. You already have a big library. Then why you want a special departmental library? Why? Because every DNA or every gene or every sequence of a DNA will not going to code for any protein. Some portion of the DNA are introns, which will be not coded by that, and that intron will be already eliminated in the RNA processing. So we will take a mRNA, and from mRNA we will make a proper DNA, which by which we will get to know which is our recombinant, which is our specific sequence or gene coding for a particular protein. So what you will do for that? Uh, for this purpose, we are using mRNA, not for the other purpose. To get and specify the tissue specific protein synthesis or anything else. Okay. So that I will produce the rhodopsin protein, but your liver cell will not going to produce the rhodopsin. Liver cell will produce the enzymes for the gluconeogenesis or glycogenesis. Okay, so this is the purpose. So by that cDNA you can make a DNA library for the eye, for liver, for bones, for the tissue, for hairs, everything. So mRNA are tissue specific, and the mRNA doesn't have the intron or UTRs. What is UTR? Untranslated regions. So before translation, mRNA have the before promoter. No, not before promoter. Before starting codon, there is a UTRs, untranslated region, or after termination codon, there will be also UTR. So here I made one diagram. This is a mRNA. This is a reverse transcriptase. It will transcribe into cDNA, and that cDNA will have a RNA strand and the DNA strand. But we don't want a RNA strand. Okay, the red color is a RNA strand. Okay, black color is a RNA strand, mRNA strand. So we want DNA only, na? So we will remove this mRNA strand. How we will remove? By adding a RNA in each. It is an endonuclease, and once it will be added, it will digest the what it will digest the XYZ. What is XYZ? It will digest only DNA, not DNA. It will digest only RNA. Understood? Then once it will be digested, it will form a hairpin, and this pin-like structure, this uh, round structure will act as a primer, and this primer will be going to be uh, it will take a enzyme polymerase enzyme, DNA polymerase enzyme, and then this hairpin will be elongated, and this twist will be cut by S1 endonuclease. Understood? So this is all about mm, mRNA and all. So now what we will do, we'll see that you made a clone, you made a clone, then how we can distinguish between that which is our clone and which is not our clone. And what is the major difference in the cDNA library and the genomic library? What is the major difference in the cDNA library and the genomic library? Could you please tell me if you are watching till now? Then you have to comment and you can download the uBiology. There you can ask your questions by clicking your question doubt and then send your question in your uBiology. Thanks. Then if you're not able to tell you now, till now, then you go and watch the video again, or you can this is what I'm going to tell you. Okay. So first is you can write a single line. In single line, I will tell you genomic library, genomic library contain both kind of books, like all books. Okay. So how I will write? This is not a book library. So I will write genomic library contain coding and non non coding sequences. Okay. Or data you can say. Okay. And cDNA, cDNA library, cDNA library contain only coding sequence. Okay. I am writing this coding in not in terms of the uh, transcription. Okay, their coding and non-coding is different meaning. But here I wish that you understand that coding means what, which will going to expressing DNA, expressing sequences. Okay, so coding and non-coding sequences. Hope so you understood this thing. Now the how you can find that is your recombinant DNA or not, or that is your 20 Down syndrome, Patau syndrome's chromosome or not. How you will find? Suppose I gave you this three, three different clones. Okay, I given this. No, I given five clones, and now there is a random sequences, and this is a random sequence. This is your clone inside this, and now I am asking, okay, Mohan, kindly tell me that among all these five, which have the 18th chromosome disorder, or which is your 18th chromosome. Okay, then how you will find? For that, you should get the exact sequence of the 18. Means first you want sample, okay, proper sample. Then only you can compare. So this is a 18 chromosome sequence. Okay. So first, what you will do? You'll you made a plates. Okay. You took small pieces from everything separately. You took this piece. You this. First, you want a sample. Sample means what? First, you want a sample or comparing sequence. What we can say that. So first you want a reference sequence. Okay, sample or the reference sequence. Huh? So now you will take. Suppose you took the sample from this, then this, then this, then this, and then this. All separately. You do one thing. What you will do? You will do. You will digest it. Okay. Took on nylon nylon membrane. Okay, membrane. Then treat with first, second, then Then treat with NOH or NOH you can treat. Okay, alkaline medium. Then what we have to do? We have to take. You took this all clones in on a nylon membrane. Then you treated with the detergent and treated with detergent. Why you want to treat? You are going to treat with the detergent, not anything else. You can treat with the acid also. Yes or no? Why you want to treat with the detergent? Anybody, please answer the question. So you have to treat with the detergent because it will not disintegrate the molecular mm, integrity of the DNA. Okay, if you treat with the acid, it will disintegrate the molecular integrity of the DNA, and then you will not get any DNA or nucleic acid. Got it? So you want to just you want to open a cell, bacterial cell. So you treat it with the detergent. Third, then you will treat with the alkali medium. Treat with alkali medium. What it will do? Alkali medium. It will denature DNA. Okay, denature DNA. Okay. Then once it will be treated, this all things is done. Then you see, I'm going to make one diagram, and it looks something like this. Okay. So this is your nylon membrane. Okay. This is your nylon membrane. I made randomly. Can you feel the colors in this? I don't know. If you know, then please comment me. Okay. Okay. You feel this. This is nylon membrane. And then what you're doing? You treat it with the DNA. Suppose this is a DNA. Okay, this is a DNA. This is a DNA. And then you will treat it with a UV ray. What you will do? You will treat it with the UV ray. Don't forget that we talked about the okay. This is a denature DNA, so you will get only a single strand. Okay, sorry, I made a so many strand. It's not a double stranded. Denature means single stranded DNA. You will get. Oh, back. Okay, so you will get a suppose here I'm making. You got it. Shit. It's slow little. Okay, if you like these videos, then you can attend the live classes on uBiology app or on Google Meets. I will be available for you, and soon we are going to update you a good information, good news about our uBiology app. So this is the nylon membrane. Once you treat it with alkali medium, then the DNA becomes a single stranded. This is your single stranded DNA. Now, what you have to do, you want that DNA should be attached to the nylon membrane. So you will treat with the UV rays. With what? You will treat with the UV rays. Suppose this is your UV rays. Okay. So once you will treat with the UV rays or baking, you can use a baking. Then this DNA membrane, UV ray helps in the DNA membrane bind. DNA will bind with DNA attached on nylon membrane. Understood? So again, you took a nylon membrane, you put on the clones from bacteria, took a clones from bacteria and put on the nylon membrane. Then you treat it with the detergent to open up the DNA, open the bacteria, and then you took what you took. Then you took a alkali medium and you after using of alkali medium, then DNA will happen. Then you treat it with a UV ray, and now you have a reference. Okay, you have a reference. This is a 18th chromosome number reference, and this is a complementary. You remembered it. This is a complementary. Then what you have to do? You put this all in a reference, and only this sequence got a complementary of this. Okay. So suppose if it is got a complementary of this genus, then if I will name these clones, this is the first clone, this is the second clone, this is the third, this is the fourth, this is the fifth. Then I will tell the second clone have 18th number complementary DNA. Okay, 18th chromosome we can say, or anything. So this is the way by which we are isolating or confirming the uh, clones which is having our interest of gene. Okay, and this technique will called as a colony or plaque hybridization. Okay, so this is my way of teaching. After teaching only I am telling you that heading, but I will improve soon. You should tell me, should I improve or it is okay? That is called colony and plaque hybridization. Okay, this is your topic. Hybridization. Hope so you understood these things. And thank you for watching till now. And in next video, I will bring you the new videos, and that will be topic will be ge- genomic markers. Okay, and the plant tissue culture. The plant tissue culture is very important topic for the IT jam and all this, because 100% three questions will become. Okay. Thanks for watching. We will meet in the next video.